Dr. Charles Lineweaver is a senior fellow of the Planetary Science Institute, researcher of the Research School of Astronomy and Astrophysics, and of the Research School of Earth Science, Earth Chemistry, of the Australian National University. He is an award-renowned expert on astrobiology, cosmology, planetology, evolutionary biology, and astronomical and space instrumentation, with dozens of published papers, books, chapters of books, conference proceedings, and articles in different magazines. So, thanks again, Dr. Charles Lineweaver, for accepting our invitation, and please take the floor.
Okay, thank you very much, Marcelo. Yeah, I'm glad to be here, and so I'm Charlie Lineweaver. You can just call me Charlie, please. Can you hear me back there? All right, good. And I've heard that there are a few biologists in the room, and so I hope you can appreciate as a biologist that when we have the Big Bang going on and stars producing these elements and then producing a planet, then somehow life getting started, that I have included in the phylogenetic tree of life the mitochondrial endosymbiosis that produced the eukaryotes over here, and then these chloroplasts, which endosymbiotically migrated into the things that we now know as plants. Anyway, this is a one diagram of the entire history of the universe and the Earth, I guess. And so I was asked to put together 4 lectures and with one rubric, and the rubric is Habitability and the Search for Life Elsewhere. Today I'll be talking about this first one, the origin of life. What is life? When is life? Where is life? How and why? Then for the next lectures, that's tomorrow, what kind of planets are in the universe, then habitable zones, and then the last lecture on Friday will be about the topic, the question, are we alone in the universe? Okay, so when I'm in the audience, I like to interrupt the speaker and say, hey, what about this? So anytime I say something that you want more details on, or you say, that's wrong because, just shout out, and I'll be happy to shout back at you. Okay, the origin of life. What, when, where, how, and why? So what is the origin of life? What is life? Well, life is what happens when you go from this cold, odorless gas to a kangaroo. And I showed this last week, and so this is like 2-slide version of the history of the universe. This, an overdensity in a molecular cloud, turned into a kangaroo 4.5 billion years later. That's what we think happened on Earth, and what we'd like to know is whether that happens elsewhere, or how similar events or processes happen elsewhere. Another way to look at that is life, non-life, evolved into life. And so we have non-life down here, and we have life over here. So if you're a scientist, you subscribe to non-life somehow evolving into life, and that's the process that we're trying to understand first on Earth, and then trying to figure out what are the most fundamental parts of that process that we could possibly extrapolate to the planets on other stars. Okay, so when did life get started on this planet? So here's the Earth, and here's now, and here's 1 billion years ago, 2, 3, 4, 4.5 billion years ago. And life got started. Life's been around for that long, and maybe even longer. And so life established itself on Earth quickly, or possibly as soon as it could have. And that's somewhere in this region here. Now, it's good to remember that about 4 billion years ago, the moon, 4.2, the moon was 10 times closer. And tides were therefore 1,000 times larger because tidal heights go as 1 on r cubed, where r is the distance from the Earth to the moon. So if the tides were 1,000 times larger, the 1 meter tides we have today were 1 kilometer high tides 4.1, 4.2 billion years ago. That's interesting, I think, for those of us who live near the ocean. So also, the rotation rate was much faster. So today we have a 24-hour day. Back then it was about an 8-hour day. But there's some argument about whether it was a 5-hour day or a 10-hour day, but basically it was a 8-hour day. So today we have 2 1-meter tides in a 24-hour period. Back then you had 6 1-kilometer tides in a 24-hour period. That's a big, big difference, particularly if you're on the coast. So the reason why this is important is because potentially this hydration, dehydration that is so much more important 4 billion years ago could have played a very important role in the origin of life. Any comments from anybody? Yes? Charlie, yes? Where does this data come from? Well, some of this data for the hours, for example, comes from geology, where you have rhythmites, for example, where you have rivers that are coming down, and then you have tidal. Well, you know, there are. You've heard of neap tides, king tides, and neap tides, where the sun is so if the sun is here, here's the Earth, here's the moon, and then also when the moon is here. So you have a king tide when they're gravitational pull. The sun's tidal effect is about 1/3 of the total. The moon is about 2/3, and then when they come together, it's in the same direction. Then you get a big tide. Now, so when you look at the tidal flooding and the mud left in them, and then go out, and then tidal flooding again, and the periodicity, you can tell the difference between daily and monthly. And that enables you to say how quickly they are happening. Also with coral, that goes back about 200 million years, you can see the layers in the coral, but it only goes back about 200 million years, or maybe 300 million. You might be more of an expert on this than I am, about using layered biology to understand the shortness of the Earth, of the day. Now, but that's kind of like using biology. You can also use just physics and say, we know how fast the moon is receding now because of the bulge. You know, if Hervé is the moon and I am the Earth, I'm rotating. If he's pulling my water this way and this way, right? But then I'm rotating every day, and he's only moving once around in a month. So I'm pulling my bulges around in this direction, and he is always pulling this bulge back, and so he's slowing down my rotation. And you can figure out how quickly that, because of something called the Q value, but you can figure out how much friction there is on the bulge, and because of that, he is receding. The moon is receding about as fast as your fingernails are growing. This is physics rather than biology, but the two come together to produce these results that I'm talking about. Okay, so essentially there were no quiet little tide pools, as this picture seems. Look at this nice little ripples, right? This is very different from 1 kilometer tide six times in 24 hours. That's very different. So when you see this picture, remember this is an artist's rendition, somebody who likes to draw pictures from Hawaii or something. Okay, so there's no oxygen, no ozone, lots of UV. So if you invent a time machine and you go back more than about 2.3 billion years, you will not have any oxygen, so take some oxygen with you. Okay, so when did life start? Well, we have to look at the history of the accretion and the bombardment of the Earth. Then we can look at the oldest rocks. Then I'll talk about the oldest fossils, the oldest isotopic fossils, and the oldest, deepest parts of the phylogenetic tree. These are all pieces of information that we use to put together to create this story of when did life get started on Earth. So the moon-forming impact was about 4.5 billion years ago, about 20 to 100 million years after the formation of the solar system. So that's about the approximate size of the pre-Earth and then this Theia object that smashed into the Earth. And just for scale, this is about the size of the impact 65 million years ago that wiped out the dinosaurs that hit the Yucatan Peninsula. This is just hand-waving, but I shrunk it to an appropriate size. All right, so here's the accretion rate of the Earth. The accretion rate in tons per million years. And the point is, this is that when the Earth got started, this is 1 billion years, 2 billion, 3, and here's today, and here's the Earth today. You can see that the accretion rate today is about a million tons per million years, so it's only about a ton per year. Not a lot. And you can see it hasn't changed much, and then you get back to here, and then you get into the danger point where you get so many impacts that you are back here. This is where like the moon-forming impact was right here. So there's 4.567 billion years ago. That's essentially the age of the oldest refractory material, the CAIs in the chondrites. And then you have 3.8 billion years ago there. This is 4 billion years ago. And so the origin of life we think was somewhere here. We're not sure where. And then the moon-forming impact was here. Notice how high this is. And as I pointed out last week when I showed this, this diagram is maybe the only diagram in the history that integrates to the current mass of the Earth. You will see this diagram of accretion rate over and over and over again, but no one has taken the time to extrapolate into the accretionary phase such that the integral of this diagram turns into the mass of the Earth. We spent some time making sure that was the case because we wanted to get this approximately right. Some people will claim that there's a late heavy bombardment here, but I'm not one of them. Okay, so the moon can be treated as a bombardometer. And look at it. See, that's got a lot of impacts. And if you look at a couple of papers here, you see this is the age of the surface, 3 billion years ago, 3.9, 4.3. And then you have the log of the total number of craters greater than 20 kilometers in diameter. So essentially, you look in a crater, you take a crater like this, and then you count all the craters inside of it, and that's used as an estimate of how old that bigger thing is. And when you do that for the moon, and then you say, hey, the Earth, because the Earth is larger, it has a larger gravitational field, and so it will be hit by more impacts than the moon. Why? Because it's gravitationally focusing these things on it. And so that's why these points are not from the Earth, because we cannot, the Earth's surface is already, because the hydrosphere has already had all these things eroded, but we can make a pretty accurate prediction that's about the Earth will likely suffer about 10 impacts by objects more massive than any that strike the moon. That's why the Earth's plot here is larger than the moon's plot. And notice it only goes back to 4.3, and so they want to keep it, they kind of restrict it to the bombardment. And matter of fact, I have a colleague who has two phrases. He says there's an accretion of the Earth, and then when the moon forms, it's solid form, then it becomes a bombardometer only after the surface cools down so it can record the bombardment. And so that's what he calls the bombardment. Before this surface cools down, well, it's still hot, you hit it and then there's nothing to record. It's just lava. It's like a magma ocean. The same thing for the Earth. And so, so what? What's the point? The point is that there are two separate words in the literature sometimes used for bombardment that can be recorded on the hard surface of the moon, and the accretion, which was responsible for the mass of the Earth, which is supposedly what I showed here. All right. Any questions about that? You guys Hervé, have you run into that distinction between bombardment and accretion? No? Well, yes, George. What about what? The late bombardment. I think the late bombardment is a hoax based on some really bad statistics and the histograms that were produced by NASA and the method in which they collected their rocks. It'll take me a while to go into the detail, but essentially what I did was we did some crater counting ourselves, and we didn't find any increase in the bombardment rate during their claimed 4.1, 4, 3.9, 3.8. We didn't see any more impacts during that interval than before or after. So I think it's, the impact rate, I think, is probably more like we drew here. That is, it's stochastic, up and down, up and down, up and down, but there's no major late heavy bombardment that we saw in the data that we were analyzing. And when you analyze the dates from lunar meteorites that have come to Earth, you also do not see it. The only place you saw it was in like 72 when they first took these histograms of the rocks that they collected, but they could very easily see what they collect a rock and a rock and a rock, and they say, oh, that's an independent bombardment, but that could very well have been just one bombardment. They were in the debris field of that one bombardment, which then loaded up the 3.8 or 3.9. That's basically the story why I don't believe it.
Can I ask a question?
Yes.
Are there any recent estimates for the exact dates for the impact that formed the moon?
I've read a few papers on that.
Can you constrain that, actually?
Well, there are a few papers on that, and I'm not an expert there. And we have, you mean this date right here. And I said right in this slide here, my information tells me that the moon forming impact was about 4.5 billion years, 20 to 100 million years after the formation, after 4.567. That's what I gleaned from the literature. It's a big error bar there, but that's based on a whole bunch of things. One thing that you have to watch out for is the body that formed, sometimes these earth scientists want to figure, when was core formation? And they pretend that the earth was there, and they say, okay, there was a segregation between the stuff, the siderophiles that formed the core, and the rocks, and the incompatibles that didn't go into the core, or the silicates. And so I forget the name, so it's a planetary science here, which, was it tungsten something? One of these elements that's radioactive goes to the core, it's a siderophile, and the other doesn't. And then they use that date as a separation of the formation of the Earth. But I'm not sure whether you call that the formation of the moon, because maybe the object was so large that it undid some of the differentiation that had occurred earlier, and then it had to differentiate again, because this obviously made the whole Earth hot enough so that you could have density differentiation again. But
okay.
It's not, I'm afraid there's the large error bar on that. And there are about 5 pieces of evidence that goes into that, none of which is good. And I've stopped my caveat. My caveat is that the core mantle segregation already happened in the objects that it created to form the Earth. So when you date the core mantle segregation of the material that is in the Earth now, you're not dating one event, you're probably dating 50 different events. And so that occurred over a period of 100 million years or so. So be careful when you hear an earth scientist who says that, oh yes, based on the segregation time of the core and the mantle of the Earth, that's when the Earth formed. Now, I think that's just a misunderstanding of the oligarchic growth period that we believe led to the accretion of the Earth. Okay. Boom, boom, boom. Okay, bombardometer of the moon. Okay, so what about the oldest rocks on Earth? Here's a nice plot of how old the surface rocks on the Earth are. In the Cenozoic here, Mesozoic, Paleozoic. I was surprised to see that the Proterozoic is 0.5 to 2.5 billion years ago. That's this pink color here. There's quite a few in Brazil here. But then the Archean rocks from 2.5 billion to 4 billion. And this is this deep red here. And so there are some deep red areas in Brazil, which, Evelyn, you're probably familiar with because you're looking for fossils in these oldest rocks here. And you were talking about the Mesoproterozoic and your fossil claims there, but that's only 1.3. So you must be in this pink area rather than the red area. So I suggest you go looking at the red area if you can. I bet you're already doing that. You're already on top of this. Anyway, there are 2 regions of the Earth that have the oldest rocks, and those are Ishua, Greenland here, and the Pilbara Craton here. And I'm going to show you some results from people who have looked at these areas. One, I'm going to show you some macrofossils of the oldest claimed stromatolites here. And here I'm going to show you some isotopic evidence for carbonates inside of zircon crystals, which are then dated at 4.1 billion years. They're not macrofossils like the stromatolites, but they are isotopic evidence for the existence of life, because it seems, for reasons I would like to better understand, that life likes carbon-12 more than it likes carbon-13. This is isotopic evidence, so I'm assuming, is there anybody who's an expert in C12, C13 ratios, makes a living out of that? The bottom line is there's CO2 in the air, and it's got a 12 to 13 ratio. There's a CO2 in you, it's got a 12 to 13 ratio, and life forms tend to have a 12 to 13 ratio that's higher than the ambient one in the gases, for example. So that's used as a lever arm to figure out when life got started. So modern stromatolites in Shark Bay. I've been out there. They have these beautiful layers here, and here's what they turn into when they get fossilized. Excuse me. And here are the oldest macrofossils. They're 3.4 to 3.7 billion year old stromatolites. Here in a 2016 paper by Nutman, here are these little domes here. And there's still some controversy about whether these are biotic or not. But I know Alan Nutman and Vicki Bennett quite well, and they're good scientists, so let's believe them for a while, but it's still controversial. Then there were previous measurements of the age of these stromatolites in the Pilbara Craton, 3.48, 3.43. So this is macrofossils. These are things that has nothing to do with isotopes. It's, hey, look at the structure. This was produced by a stromatolite. Now, Evelyn, I think you said to me earlier that you were convinced that these were, in fact, you do. So you're shaking your head. There's Abigail Allwood at JPL is the person who thinks that, no, these are not, somehow you've misinterpreted. But in any case, like any field at the frontiers of research, there's always disagreement. That's healthy. But if you want to know more details about why Nutman is right, talk to Evelyn, who's a local expert in stromatolites. Moving on. Oldest microfossils, notice this is older, 3.8 to 4.1, is these carbonate granules inside of a zircon. The zircon can be dated, and then if you find carbon inside of those zircons, then that gives you the age of the carbonates. And then you can check for the 12 to 13 ratio and see if the 12 to 13 ratio is representative more of inorganic or organic carbon. And so therefore, organic carbon with a low 13 to 12 ratio can be evidence for life. This was first used by Moiges in 1996 for 3.8 billion. And then in Bell, more recently, 2015 at 4.1. It's pretty much the same technique using the 12-13 ratio as evidence for I have found life. So here's the plot in the Bell paper. So here's the high 13 to 12 ratio. Here's low. So here's organic carbon. This is what our bot, I guess I'm minus 20, you might be minus 25, I have no idea. But anyway, this is where life is, and this is where the organic carbon is. That's where you will find CO2 in the atmosphere, I'm guessing. That's what I've been told anyway. Anyway, so just to give you another more detailed, so these are the hunters of low 13 to 12 ratios, Moiges, McKeegan, and this paper, Bell et al. 2015. And here's their result, is minus 25, and at 4.1 billion years ago. And just to show you a more detailed estimate show you how variable life is. So here are life forms, and here is marine carbon. These are the inorganics, and here are the organics. You can see there's quite a variety here. Also notice that the methanogenic bacteria are way, way down here, minus 85. That's probably about right here. So there's quite a range. It's not a done deal. It's not black and white, but you should be aware of the range. And here essentially is what life goes much further, and non-life is up here. And that's the degree of the security of this 12 to 13 ratio as isotopic evidence for for the origin of life, for the evidence for the origin of life. What about, so I should stop there. And does anybody have any questions about that particular? This is the most important, this is one of the most important pieces, maybe it is the most important evidence for when did life get started on Earth, because this is the earliest evidence. Notice that this predates the stromatolite evidence by about 500 million years, or at least a few 100 million. Gustavo, question? Gustavo has a question, Marcel.
Could you use this carbon 12 to carbon 13 range to deduce the somehow phylogeny of life?
Well, that's essentially what was done here, right? I mean, C3 plants and C4 plants. See how different they are?
So they are certainly younger. And what's the largest range that you see?
Well, it goes down. It goes to methanogenic bacteria, minus 485. That's about over here.
Okay. So you don't see as large a range in, say, eukaryotes and more modern life. You don't see this range. I
do not know that. We haven't. No, these are eukaryotes right here. And these are eukaryotes. Here's eukaryotic algae. Here, bacteria. So here are the eukaryotes right here, and here are the bacteria. So the bacteria typically have a much broader range of 12 to 13 ratios consistent with being them. And typically, eukaryotes are monocultures. So that you're seeing that in my miniature version right here.
Okay.
Any other questions? We must have a biologist. Is there any biologist here?
I have a question.
Okay, go ahead.
Yeah, but it's not clear for me why the life prefers the carbon 12 and not the carbon 13.
Okay, I've asked that question to people who should know it, and I've gotten 3 or 4 different answers. 2 answers are when you're holding on to something, and 13. If you have a heavier nuclei, you don't vibrate at as high a frequency. So when you make a bond, you're kind of vibrating, right? And if you have a heavier nuclei, like 13, you don't vibrate with this high a frequency. That makes the bonding strength lower for the 12 and higher for the 13. So that's just kind of like swinging around something, and the more mass it has, the more stable it is. And if you, it's a kinetic effect. Now that's one suggestion. Another suggestion is, well, if you make a box with a bunch of, let's say, protons and deuterium nuclei, so protons and then twice the mass, and then you put a hole in it and heat it up to a certain temperature, you will have more protons going through that hole than the heavier ones. Why? Just simply because they have a higher velocity. They're both at the same temperature, therefore there's a higher velocity for the smaller ones, and therefore they're going through that's another explanation I've heard. There might be two more, but I'm very dissatisfied with the experts who should know more about this. But you should ask your own local, I don't know, quantum chemist or biochemist, or I'm not sure which exactly field this would belong to. But I just told you the results of my survey of people who should know.
These two you mentioned are the same, basically.
Say again?
I believe both examples that you gave are basically the same. They're kinetic, inertial reasons, basically. In biochemistry, this is very common, why some metals in proteins are substituted by others, and that changes the whole thing just because of speeds, kinetic reasons. So.
Okay, so the big question is, do you think, if life elsewhere is based on carbon, do you think that there will be a preference for that alien life for C12 over C13? Is it really a universal feature of life elsewhere or not? I don't know, but it seems to be a feature of life on Earth, and that's what was used to get this earliest evidence for life on Earth, and that's why it's so important. Okay, where did life get started? Well, a few years ago, we did this plot. You make a phylogenetic tree of life on Earth, and you have to culture these things and then figure out what temperature tolerance they have, and then you color code them according to the temperature. And here's the maximum growth temperature. This is 60 degrees, 70, 90, and then 100 or more. So, you know, when you're a eukaryote, so when you take a shower, your shower will always be less than 60 degrees. Otherwise, you'll denature the proteins at your surfaces on your skin. Now, that's why all of the eukaryotes here are in green. But there are many, many others, as you know. There are thermophiles and hyperthermophiles, and they like heat. And the point of this diagram is to show that the organisms with the shortest distance to the root are the hyperthermophiles. That suggests that the origin of life was hyperthermophilic. This, too, is a contested statement, but the data that I've seen, I think it's pretty good that hyperthermophiles were at the origin of life. So that means life started out hyperthermophilic, then it evolved to be thermophilic, and then it evolved to be mesophilic, and we are mesophiles. There is some controversy about that. So the two models for the origin of life, if you want to know how life got started, there are two very common models, and that is hot hydrothermal vents that are at these places here where the seafloor is spreading. And then there are the hot springs. So these are essentially volcanic something or other that come up and heat the, this is like prismatic in Yellowstone, where you have a volcanic active site, kind of like in Auckland, around Auckland in New Zealand. But there are many places. Are there places like in Brazil where you have active volcanism that's heating up freshwater pools? Not that you know of? Where is that? Where? Central Brazil. So a volcanic area, hot springs. Okay. Okay, anyway, so those are kind of, the reason why I call them two different models is because although they both depend on heat that's coming from the earth, well, one is freshwater and one is salt water. So hydrothermal vents, here's a blow up of that previous slide, and here's where they are. And so here we have heat from mantle convection rises at the mid-oceanic ridge spreading center. And that spreading center is responsible for making South Africa go away from Africa, dividing that about 120 million years ago or so. Anyway, there are black smokers that are hot in this hotter region, and then off the axis a little bit are white smokers. They have warm water vents, 20 to 50 Celsius. And the reason I mention these two is because These are alkaline, and these are more acidic. And the people who study and make a living out of hydrothermal vent scenarios for the origin of life prefer the white smokers to the black smokers. So here's well, the difference is the heat. For one is the temperature difference. These are much hotter. These are cooler. You see the temperatures? And the other, these are like iron, magnesium. These are alkaline, and I believe these are acidic. The ocean water is about a pH of 6 or so. This is pH of 9 coming out of here. So there's a pH difference that is probably the source in this model of the energetics of the first life on Earth if it originated at these alkaline hydrothermal vents. And I'll show you a little bit details of that. Okay, so here's what a white smoker at a hydrothermal vent looks like. You can see it's white. It's not black. And now, interestingly, there's a paper by Weiss et al. This is Bill Martin's group in Germany. He talked about the physiology and habitat of the last universal ancestor. And Bill Martin's group is somebody who, I guess, is convinced that the white smoker origin of life is the best one. And in this physiology, the physiology of this common ancestor, it's anaerobic. It has to fix CO2. It depends on molecular hydrogen. It has to fix nitrogen, and it's thermophilic. And those are the features of their, I guess, their analysis of the deepest physiologies in the phylogenetic tree. So here's a plot from their diagram. This is Weiss et al. 2016. Basically, you have the ocean here, and you have hydrothermal fluid here. This is all hydrothermal fluid. This is the ocean. There's some kind of membrane here. Then you have a pH. Notice the pH difference. This is the most important thing if you're interested in the redox, the energetics of the origin of life. pH of 6, pH of 9. So that means there are more protons out here than there are here. That means there's a proton gradient. If there's a proton gradient, that can be a source of energy. So if you have anything here that can, I guess, tap into the energy, it's kind of like having a water flow. You have a water coming downhill, and you'll have to put a water wheel in there to do some work. That's what this represents. These are, first of all, starts geochemistry. Then you have this pH difference. And then you have a membrane transport protein, and the deal there is kind of like a water wheel that you put into a stream that's going downhill, and that is when the proton difference, that you'll naturally want the protons will fall in like this, and then that energy is used to get rid of Na+, for example. So that's how, these membrane proteins are hypothesized as the first, essentially, the metabolism of the earliest life. Essentially a proton gradient, falling of water that you stick a water wheel in, and then it makes something go around. In other words, it makes you able to pump things preferentially in or out of your membrane. And then you have an ATPase or an ATP synthase, which they have here. Now, this is interesting. They have here a sodium fall because you pumped out the sodium so you can make the sodium come back in, and then you make ATP out of ADP by adding a phosphate. And but when I looked up ATPase or ATP synthase, it's always a proton coming through here. And does somebody know more about this than I do? Why do they have sodium? I didn't know there were ATP synthases that used sodium gradients rather than just a proton gradient. Anybody want to chime in here? Anyway, that's a topic that you could look up yourself, I guess. Okay, so moving right along, let's see. So life on Earth could have started in places similar to the Grand Prismatic Hot Spring in Yellowstone National Park. It's freshwater, not saltwater. And these are wonderful colors from the different types of bacteria. And they could call it Darwin's warm little pond. Notice it has UV. Notice there's no UV whatsoever at the hydrothermal vents because they're under 2-3 kilometers of water. You don't get UV through that amount of water. Here at the surface, you do have the UV. Why is that important? Because UV radiation, as you know, can take electrons and pump them up, and they can energize molecules, which can then do something interesting because they have been activated. Whether that activation is important in the origin of life or not, we don't know, but the proponents of the surface origin of life take advantage of that UV to explain some of the weird chemistry that you can do once you have activated molecules and atoms. So here's a diagram from this group. This is Bruce Damer, Dave Deamer Van Kranendonk and also Dimitar Saslov. These guys are the people who are pushing this model. And this is a diagram from their paper. So the bottom line is fresh water, not salt water. And the wet, dry hydration is by evaporation, not the tides that we talked about earlier. And I don't know how you would have wet, dry cycles at the hydrothermal vents. Some people said that rules that model out, but other people say you can have that at hydrothermal vents. It's just a question of the concentration of the water in the fluids around the chemistry that you're interested in. Does anybody have anything to say about that? Okay. All right. In any case, you have UV radiation, and here's the thing. You have essentially organics coming here. They're falling down here. There's evaporation and evaporation, drying and cooling, drying and wetting, and then you get these bags, and then something interesting gets left over, and then they adapt as they get closer down to the mountain, into the ocean, I guess. Yes?
Would you agree that the hot spring model provides this UV activation which is completely absent from the hot vent model, but then the hot vents probably they provide more stability. And you wouldn't say that you need some kind of very long-term stability for these chemical processes to really establish themselves? What do you say?
I don't know. I don't know whether stability or excitation is more interesting. For example, another example, there was this gigantic bombardment during the 4 billion years ago. Was that bombardment destructive, or did it frustrate the origin of life? Or do you need that type of hot chemistry to have the origin of life? Another example is Jupiter. Is the presence of Jupiter, did that protect the early Earth from bombardments, or did it cause it? And so there are all kinds of questions like this, and we don't know I don't know the answer. I don't think we do either, of whether it promotes, whether Jupiter had a role in promoting bombardment or protecting us, whether UV has a role in destroying life. We heard people talk about, oh, UV, if we get rid of the magnetic field of the Earth, we have more cosmic radiation, and so maybe that's dangerous. Or on the other hand, maybe that increased the mutation rate to a level that is needed. So these are big questions we don't know the answer to, but we're investigating them.
But would you agree that this step-by-step complexification needs some kind of stability for it to work out? Yes and no, because suppose that bears hibernate. And, my point is that life can find ways to stabilize its environment to go, like, for example, I was out in the sun, and if I went, oh, this is too hot, what do? I just go in the shade of a tree. Now, if life has that ability, then it's self-stabilizing, and any, okay, you're going to turn off the sun, and the sun goes out behind a cloud, I come out. So life is like that. Life is self-regulating, and as long as if you can turn that on, then the variability in the environment is not the main factor that you're dealing with. But you have to acquire this ability, this kind of teleonomy at some point, and you have to get there. It's not easy. Well, that's essentially what life is. Life is the acquisition of the ability. So maybe, I would say it's all spandrels, and what that means is that you acquire it by accident. And so that's the way life had to have emerged, by some accidental thing that turned into an adaptation. And those are the ones that survived, and that's why we call them life.
Just a comment on this. In cybernetics, Ross Ashby's idea there is for life and applies, we have stability and ultra-stability. It's a nice distinction. Stability is the basic system when you survive in normal resources, and ultra-stability is when you cannot adapt and you are in risk of collapse. Then you go to another state like stasis, dormantia, hibernation, this coma, fever. There are all kinds of...
Right. Another aspect of the same question is, do you need 3 phases for the origin of life? Do you need gas, liquid, and solids? You could say solids, well, that's the atoms are kind of like this, and there's not a lot of interesting chemistry that goes on in solids. In gases, the atoms are going all over the place, so nothing is stable. But what is liquid? Liquid is when you get something interesting passing, and they're kind of like metastable. They can hang on to each other, and therefore, maybe liquid is needed for life,
because of that metastability of the liquid state compared to the overly stable solid and the underly stable gas. It's the same, pretty much the same argument. Anyway, I'll move on. So 2 models for the origin of life, hot thermal vents and hot springs. Here you have salt water, no UV, no atmosphere. So here you have a liquid solid model. And here you have a liquid solid gas model. 3 phases, you have fresh water, UV, and an atmosphere. All right, but also maybe life got started on Mars. So far, these 2 scenarios happened on Earth. But maybe the same or similar things happened on Mars, and then we had a bombardment, and then bacteria came to Earth coming through a Mars meteorite, and then it started to evolve. It said, hey, here's a nice place too. So that's also a possibility that shouldn't be ignored. And you can see that there were very vast flows of water on Mars 3.5 billion years ago and earlier. Okay, also one thing to keep in mind is what is heating the Earth? What about the center of the Earth is hot, the surface is cool, and then you have this volcanism. Well, you have there are these 4 radiogenic elements that are producing heat, but you also have the heat of accretion. So the Earth had to come together, and then when things fall down, they produce heat. But also you have these calcium-40, uranium-235, uranium-238, and thorium-232. Here are the different half-lives, and here's the total heat flux from these 4, and you can see it goes down with time. And here we are today at about 20 terawatts, and here we are at 4 billion years ago, about 80. So it was 4 times more radiogenic heat that was being produced 4 billion years ago, and that's the period we're interested in, because that's when life seems to have gotten started on Earth. So that means there were a lot more volcanism and a lot more hot springs around the territory, around the whole surface. And remember, the Earth was also rotating with an 8-hour day, and so you had these 1-kilometer tides. The moon was on the order of 10 times closer. So this is a very weirdly different, much hotter region that is our best estimate of the environmental parameters 4 billion years ago that constrained whatever it was that led to the origin of life. I should say all these physical parameters are ones that I have no hesitation in extrapolating to the other Earth-like planets that we've detected so far in the universe. None whatsoever. The ratio of these radiogenic elements are going to be within a factor of 2 on these other Earths. They're going to be rotating because of angular momentum that involved in a protoplanetary disk formation and the formation of planets. All of these things can be very similar to what happened on Earth. Some of them can have large moons like ours, or they'll have smaller moons. They'll have more water or less water by a factor of 2 or 5 or so. Anyway, there's a lot of variability, but these are physical variables that will have a well-described, or pretty well-described, distribution function that we can throw at it and say that's probably a good description of all of Earth's. And so these are not unique features of the Earth. Nothing that I said here is unique to the Earth, or should say, oh, this could only happen on Earth. For example, imagine if there was some really weird element that we had on Earth we didn't have anywhere else. Then we say, it's responsible for, and is directly connected to our theory of the origin of life. Then we would have reason to talk about Earth being rare in terms of its ability to produce life. Did I say something? Okay. So the origin of life. When did life get started? Well, before 3.5 billion years ago. 3.7, 3.8, 4.1, we're not quite sure. But it was probably after the moon-forming impact. And maybe life originated several times. We've written a paper, Paul Davies and I have written a paper about the second origin of life and how biologists may not be able to detect it. Maybe it still exists now, but because we keep on looking for the same templates in RNA and DNA that we're not able to find it. But maybe we will find a different type of life on Earth. Not too dissimilar from the way Carl Woese, about 40 years ago, discovered archaea. He discovered a new type of life because what was that? It was a branch that was deeper in the tree of life. And so that was kind of like a second type of life. Possibly another thing will happen if we get better and better at sampling metagenomics. Okay, so when this time? Where? Here, or possibly Mars. So there's an interesting question. Why isn't life starting now? And Charles Darwin speculated about this. Here's a quote from him. He said, "It is often said that all the conditions for the first production of a living organism are now present, which could ever have been present. But if, and oh what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes, at the present day such matter would be instantly absorbed, which would not have been the case before living creatures were found. I call this the Steve Jobs argument, because you cannot reinvent a computer now, because if you did, people would just start laughing at you and say, we've already done that 50 years ago, and you will not be able to form a company, and you will not get successful. Why? Because it was already invented. That's the argument that they're invoking here. But I'm not quite convinced. If there's no, like he says, I believe that this was probably the case, that the conditions now are somewhere on Earth similar to the conditions that led to life. But if this is the case, they would, he says, would be instantly absorbed. I think maybe he's talking about instantly eaten by some type of, not a phototroph, but a heterotroph. And so maybe more effort should be gone into, I know there's only one scientist I know of, Dave Deemer, who's gone to places where he thinks life could get started and is trying to sample to see what it is that is doing this absorbing of these proto-life forms, because it seems to me that this is a project that somebody could do around here, try to figure out, okay, where would life have gotten started? Let's figure out the physiology of the organism which is doing the absorbing of these proto-life forms, and then maybe, I don't know, do an exclusion experiment, get rid of that life form, figure out how to kill it, and then see if these other things evolve a little bit more. Just an idea. So what is life? What is life? This is most people think they know what life is, and I think we should stop pretending that we know this. Our animistic ancestors thought that being alive was a fundamental, irreducible property of nature. Therefore, there was no what is life problem. There was more of a what is non-life problem. So that reverses our usual conception. And is life just whatever biologists study? If you talk to biologists about what is life, you're talking to the wrong people, because they really do not know. And that's interesting that people who study the thing most intensely do not know what they're studying. But I say that based on this survey. I went around the world surveying biologists and asked them, are viruses alive? A quarter said no with confidence. A quarter said yes with confidence. A quarter said I don't know with not much confidence. And then a quarter of them said it's not an important question. So these are the most abundant organisms on the planet. They outnumber bacteria by at least a factor of 10. They do things, and the existence of viruses is good evidence that there is no well-defined boundary between life and non-life. For example, when we talk about an RNA world, for me, that's a viral world. And then I say, what's the metabolism for this RNA world? And people say something like, oh, there's redox gradients or so, that the concentrations of phosphates or something. Well, that means you can have viruses that are dependent not on previously existing organisms, but on phosphate gradients. Anyways, I throw this at the biologists and say, I'm happy to report that we do not know what life is. But that doesn't stop people from trying to define life. Aristotle, for example, says, of natural bodies, some possess life and some do not. That's a black and white version, where by life we mean the power of self-nourishment and of independent growth and decay. Now, every time I see this word self-nourishment, I say, this is so silly, so naive. Self, what in the world does that mean? So if you took a male and a female student from this room, hermetically sealed them in a capsule, put them into outer space, there would be zero self-replication, self-reproduction. Why? Because every life form on this planet is dependent on the other life forms. We're all mutualists. For example, right now, what are you doing? You're breathing oxygen. Did you produce that oxygen yourself? No, you didn't. That was produced by other organisms. 20% of the atmosphere is oxygen because of these cyanobacteria and the plants. So we are dependent on them. We are parasitic on them, or we could say parasitic, but we like to say, oh, the viruses are parasites, not us. Now, viruses have outsourced protein production to other life forms, cellular life forms. Does that disqualify them? That makes them, I would have thought that makes them smarter, better able to deal with life, better able to deal with the environment. For example, let's suppose that there's a giant asteroid that comes and wipes out all the, I don't know, lots and lots of life. Viruses and bacteria are going to be here, but we will not be. So I don't know, maybe they've found a more robust niche that is to these types of problems. Anyway, definitions of life. Here's the one used by NASA the most: self-sustaining chemical system capable of Darwinian evolution. Self, again, I don't know what he means by self-sustaining. All life forms are open. They require a redox gradient from somewhere else. If you cut that off, they die. It's not self-sustaining. It's just crazy to use that word again and again. It's stupid. Now, Darwinian evolution is another word. Now, here's a question. Did Darwinian evolution evolve? Because this definition says, as soon as you get Darwinian evolution, then you have some life. But where did Darwinian evolution come from? And so you have to take that apart, and probably it had a little bit of this, a little bit of that, then it joined with something else, and then, presto changeo, it is what we now call Darwinian evolution. At least that's my version, my hand-waving version of the evolution of Darwinian evolution, which you need in order to... So there's two self-oxymorons here, this one and this one, I think. Anyway, read Eric Smith's book. I can recommend it, 2008, A Chemical System in Which the Flow and Storage of Energy are Related to the Flow and Storage of Information. Now, here's Jack Szostak. I have a lot of respect for him. He's got the Nobel Prize for telomeres. But in the last 20 years, he's been working on trying to do a synthetic origin of life in his lab. And he wrote a paper in 2012, Attempts to Define Life Do Not Help to Understand the Origin of Life. And I agree with that statement. For example, here are a bunch of things. Are there size boundaries to life? So here we have multicellular organisms, unicellular organisms. We're comfortable calling that life. But is an ecosystem life? Is endless arguments about Gaia and whether the whole biosphere is alive or not. And then you could go down to the size scale. Here, are these too small? Is a virus too small to be alive? Like I said, about a quarter of the biologists say, no, it's not alive. Then you have things smaller. Then you have genes. Then you have jumping genes inside. Are those jumping genes inside of you alive? Are the mitochondria inside each of your cells alive? They used to be alive, and now they're kind of a mutualism with us. And so these are, I think, fundamental, some people call philosophical questions that you have to address if you have any hope of extrapolating your notions of life on Earth to another planet. And that's what astrobiology is trying to do. Is life in unchanging platonic form like the number pi? Does it have a stable, unchanging definition? And my friend Friedrich Nietzsche says, 9. No, it doesn't. Only that which has no history can be defined. This is what he said in The Genealogy of Morality. So I'm a Nietzschean in this sense. And I would say life has a history, therefore it cannot be defined. A simple example of that is, you say, okay, eyeballs. Eyeballs can be defined, right? I have an eyeball, you have an eyeball. Let's define them based on what we understand eyeballs today. You go back in your time machine, 300 million years, you will find a proto-eyeball. You go back another 300 million years, you will find a proto-eyeball. Go back another 300 million years, you will find a photosensitive pigment that no one today would ever call an eyeball. Go back another 300 million years, you have photosensitive pigments that I don't know what they do, and they would not be eyeballs. The point is that something that is well defined today, eyeballs, gets deconstructed as you go back in time because it evolved. That's the nature of eyeballs, that's the nature of heads, that's the nature of life. And it may be the nature of Darwinian evolution. So in the Joyce definition, capable of Darwinian evolution, you think there was a black and white thing? Okay, no Darwinian evolution, boop, Darwinian evolution. No, I think the best way to understand these is to understand the transitions and the complications of this transition to what we now call Darwinian evolution. Just like if you want to understand an eyeball, you don't define it. You try to understand how it evolved to be where it is, because in another 300 million years, it won't be like that anymore either. Probably, or be some modification of that. Also, if you haven't read this book, What is Life? You should. These are 1943 lectures given by Erwin Schrödinger. And this is something I want to end this talk with. He said, everything that is going on in nature means an increase of the entropy of part of the world where it is going on. Thus, a living organism continually increases its entropy. What he means by that is its entropy of its body, but also the entropy of its environment. What an organism feeds on is negative entropy. The essential thing in metabolism is that the organism succeeds in freeing itself from all the entropy that it cannot help producing while alive. So this is more or less a thermodynamic argument that should be friends to the physicists in the audience. And this is something that was promoted by Ilya Prigogine. Here he is. And from a thermodynamic point of view, living systems are an instance, a subset, of a more general phenomenon of far from equilibrium dissipative systems, things like hurricanes, whirlpools, fire, and convection cells. And here's a hurricane, here's a galaxy. They are dissipating entropy. And here, does anybody, any non-astronomer know what this is? Hervé, do you know what that is?
I know what it is.
Oh, okay. Anyway, it's the surface of the sun. These are convection cells. Each one of these convection cells is about the size of the moon. And that's because why? It's because it's hotter underneath and it's cooler at the surface. So heat is being transferred faster to the surface of the sun than would be the case if it had to rely on diffusion. So convection cells are a way to undo a temperature gradient faster than would be the case without that complicated convection cell structure. That's why it's called a far from equilibrium. That means a convection cell. Look at that organized flow of material that otherwise shouldn't be organized. It's just, but here it is, a gigantic organization everywhere. And what is it doing? It's producing more entropy than would be the case if it didn't exist. So here's another example. It's a cool example. This is like a photograph of the first atomic bomb ever to go off. This was done by this guy here, and the shutter speed was like a millionth of a second or something. And here are these really weird structures here. So I saw this and I said, well, what are the purpose of those structures? I don't know, but I bet that they're there to increase the entropy because you have a lot of heat that needs to get out, and you don't have time to form a convection cell, but I don't know what those are, but I'd love to know. But I don't. But they are structures. They're far from equilibrium, obviously. And I bet at least $2 that is the purpose of these things, or what it does is to increase the entropy faster than would be the case if you didn't exist. So here's another life form that's increasing the entropy of the universe. And we got a whole bunch of things here doing the same thing. So as you breathe in, breathe out. Remember, you got to keep the CO2 cycle going. Breathe in the O2, put out the CO2. The CO2 goes to the plants. It goes around in a circle. Anytime you have a circle that maintains structure that is far from equilibrium, you have to produce more entropy to maintain that cycle in those structures. For example, if you're a college student and it gets hot, and you have your refrigerator inside your room, and you do not have a vent to the outside from your refrigerator, you try to open up your refrigerator to cool your room, it will not work. You will heat up your room, and you will heat it up more than if you didn't do anything. Why? Because all that electricity is going to cool some area, but then it has to it's not 100% efficient, and so you're heating up your room. And that's whenever nature produces a structure, that's exactly what it's doing. It's producing more entropy to keep you at low entropy, and what's the net effect? Produce more entropy in the universe. So why is there life? I think entropy production. And far from equilibrium dissipative systems exist in order to produce entropy. And who says you can't put teleology into an astrobiology talk? So from a thermodynamic point of view, living systems, we showed you this earlier, but the little meme that goes with this is entropy is the price of structure. And I saw this and I liked it, but then I said, what's something wrong with that? Here's what I think is wrong. Entropy is the reason for the structure, is more correct. And if you're interested in this, there's a paper 2008 called Life, Gravity, and the Second Law of Thermodynamics, in which at the last sentence of the abstract says, this represents a paradigm shift from we eat food to food has produced us to eat it. So have a look at that paper if you're interested in more of this idea. And I should point out that some of these ideas that I just went through are in the website arewealone.us. Well, how did we get here? It's a website, video-based MOOC, that you can have a look at if you want more of me than you're getting here. And that's the end. Thank you very much.
Thank you, Charles, for this very nice lecture. So, questions? Don't be shy.
How does the entropy paradigm adjust to the apparent fact that life arose in a thermophilic, not a maybe thermophobic environment? In the case of the formation of the main molecules, the first complex molecule.
The thermophilic predilection of early life has to do with the available redox gradients due to chemical disequilibrium, which are concentrated at these areas where there's the biggest temperature gradient, and that is at these hot springs and hydrothermal vents. So you have a temperature difference, the fluid comes out, it's quenched, it's cooled rapidly, chemical disequilibrium, then it's kind of like a redox structure. It's kind of like having a fresh battery. There's the battery over there, so you grab the battery, and then that's presumably the first energy source for life. That pH gradient that I pointed out that gives you the hydrogen gradient, that is also a result of this temperature difference. So we have a pH 9 versus a pH 6. That gives you some protons that can go through a membrane, and you can tap into that. Is that what you're asking? Does that answer your question? Okay.
Okay, so another question?
Oh, congratulations on the wonderful talk. Very challenging. And you mentioned something about emergentism. En passant, the other week you always talked about this. You have a criticism on the concepts of emergentism, because there is epistemological and ontological. So there are different ways to approach the subject. The emergence of laws that only exist when the substrate, the material substrate, emerges. So atoms do not perform catalysis, or at least reproduction or concepts of biology. And proteins cannot evolve in the Darwinian sense, because they are no complex subjects that produce populations, and Darwinian evolution is a population stuff. It's something acting on a diverse forming population. So when you have cells and you have different populations with differences in the genome, you can talk about Darwinian evolution. So in a sense, this emerges. It emerges when the substrate is there. And it is not the case that it does not exist before, because the substrate doesn't exist, so why talk about something, no?
Yeah, okay. I'm not sure I can directly answer that question, but this reminds me of an argument that I have had with quite a few people. Matter of fact, with Elisa last week. And the question for me is, we all have genomes and have information in those genomes. The question is, where did that information come from? And I kept on saying the information in all human genomes and all the information in life came from the environment. What do I mean by that? There's architectural information. There's redox gradients, hot and cool, and hot, low pH, high pH. If an environment, suppose the environment were perfectly uniform. In other words, I'm a cosmologist, and so we know the early universe was a hot, hot plasma at the most accurately measured black body temperature ever. It's 2.725 Kelvin is the temperature of the cosmic microwave background radiation. That is more or less the definition of equilibrium. If you are any type of structure in such an environment, there is no information in this black body radiation that can put any information into you. So all of the information in you is because you had one ancestor who was blind, and another ancestor had photosensitive pigments, and this one survived. You had one ancestor who could deal with the temperature variations, you had another who didn't. So those variations, select, that's why it's called natural selection. The natural environment, the information in the architecture of the environment is what did all the selecting, and that's what put the information into all the genomes. That's I call that just, it seems to me that makes common sense, but Alicia was pointing, she didn't like that argument at all. She wanted to think that the information is in the genome, and then somehow there's selection. And I said, there's no information in any genome until you have selection, and then you get the information.
But don't forget, don't forget that basically evolution acts in populations, modifying phenotypes, not genotypes. The surviving organisms carry the genotype that will be really selected in this process. So evolution does not act upon information stored. It acts upon things acting. Proteins acting is not enough because they do not characterize as organisms, but cells really do.
I think the phenotype-genotype distinction that you're invoking here is not as relevant as it is to most of biology, because we're talking about the origin of life. I think that, so, you know about ribozymes or DNA enzymes. These are, information carrying molecules that also can catalyze, right? So there you have an example of, and that has been used by Czech ever since 1980. I think he found these as an example of undoing this chicken and egg problem. It's a genotype-phenotype undoing. So I don't think I think you need to forget about genotype-phenotype differences when you're trying to understand the molecular evolution. Okay. Now, this fellow in the red striped shirt here, you were nodding a couple of times in agreement. Do you have any questions for my talk? No. I'm done. No questions. Okay, no questions.
Any question from the students? Please, don't be shy.
Yes, students, please don't be shy.
Yeah.
You're allowed to think.
I have maybe a silly question.
Okay.
About again about the carbon 12 and carbon 13.
Yeah.
So it's funny because the ratio of carbon 12 and carbon over carbon 13 is also a stellar age indicator. So if you have a low carbon 12 over carbon 13 ratio, this means that the star is evolved.
Wait, stop there. I wasn't aware that we had good estimates of stellar isotope ratios. Am I mistaken?
No, we have some, for some stars we have the method called carbon 13 footprint.
Really?
That's possible, for example, to identify supergiants in a sample of young stars, for example. So if you have a young star, the carbon 12 over carbon 13 ratio is higher than for supergiant, because the supergiant can produce carbon 13.
Right, but you're telling me that you have spectra that are sensitive enough to distinguish carbon 12 lines from carbon 13 lines. Exactly. How about helium 3, helium 4?
No, not helium 3 or helium 4, but carbon 13 and carbon 12, yes, it's possible. Because you can 16, 17, because actually you cannot see the carbon 13 and carbon 12, but you can see the CO with carbon 13 and the CO with carbon 12.
Right, but that isn't necessarily the star.
Yeah, yes. But my question is the following.
Okay.
Because I'm thinking about the enrichment of the maternal cloud that creates the sun and also the planets. So if you had a contamination from the evolved stars, and those stars were evolved and produce a lot of carbon 13, so you have an environment with less carbon 12. So do you think that this can be a problem for the origin of life? Oh, no.
I don't think so. I think that no matter what your carbon 12 to 13 ratio is, it could be 5 to 1 or 12, 13, a lot more 12. It's like 10 to the minus 5 or something, or what is the absolute ratio of 12 to 13 in like the air we're breathing?
13 in the interstellar medium is something around 90, and then
90,
drops to 20.
So for every 90 C12, there's 1 C13?
Yes.
Okay. So let's suppose that that's the case here on Earth. Now, and then we prefer the 90, the 12. Now, let's suppose that what you propose is different types of pollution of the environment, different amounts of evolved stars, and let's suppose that ratio, I don't know, was 200 to 1, or maybe it was 20 to 1, right? If we had a good explanation for why life prefers 12, that still would be irrelevant about the absolute levels or the value of that ratio, and so I imagine you would still see this 12 to 13 signal for life. But that's just waving my hand.
Okay. Thank you. Another question?
Thanks. Hi. It's a very, very, very interesting talk. I just had one question about your liking the definition that you gave us life as a producer of entropy. And you showed the convection bubbles in the sun and showed galaxies and so on. Isn't there some qualitative difference between the two in the sense that to produce a convection zone, I don't need the information there is in the convection zone or a galaxy itself, given the external conditions that appears. In the case of life, the information for the memory of it is very efficiently passed through time. Isn't life, differently put, a very efficient way of preserving information, memory?
Yes, I would agree with that. I do have a colleague who, but there's no reason why that, like you have, I don't know, 100 trillion cells in your body, and each of those has the pretty close to the same 3 billion base pairs. And you could ask yourself, wait a minute, if I only need like 5 genes to make a fingernail and another 5 genes to make liver cells and another 20 genes to make a stomach or something, why do my stomach cells have all this redundant? Why isn't it a need-to-know basis? So you mentioned the point that you're saying is that life is a good accumulator of information. And my colleague said, well, wait a minute. You can have information, as I pointed out earlier, you can have information in the architecture of the universe, and that puts the information into the DNA, right? Now, but consider stars. Stars started out with almost zero metallicity. And then what did they do? They go boom, and then they spread metals all over the universe. They spread O and C and N. And then what happens again? They have the big massive stars that go boom. Now stars are usually born in stellar nurseries. And those stellar nurseries are then polluted at a certain level by the previous generation of stars. The level of pollution then changes the IMF, the initial mass function, because when you put in some metals that can more easily get rid of radiation from the centers, I mean, the whole point of a star is to get the radiation that's formed in the middle out to the surface. If you have lines that are very rare, you can do that because it's more transparent to those lines. That's why you, when you have C, N, and O in a star, you can then get that radiation out. So what I'm trying to say is the presence of the pollutants of your parent generation can determine the characteristics and some characteristics of the next generation of stars and so on. But that information is not really in the stars themselves. It's distributed in the architecture, in the elements out of which the next generation of stars form. So in that sense, you are accumulating information, but it's not inside a cell or anything, but it is architectural information that is evolving and determining what the next generation of stars will be. And in a hand-waving way, you could then say, well, stars are doing something similar to information that life is doing.
Yeah, I would not put exactly as accumulation because you can evolve some animal to minimum information as time passes. But I'm just saying memory with high fidelity. Life would be a very efficient way of keeping memory of high fidelity.
Well, I think that high fidelity reminds me of arguments about species. I'm not a high fidelityist because at the origin of life, so much was associated with horizontal gene transfer. And if something is not low fidelity, I mean, horizontal gene transfer is like the apex of non-fidelity. You're importing a whole plasmid to change your whole metabolism, and that is not fidelity. So to the degree that horizontal genes transfer was involved at the basis of life, that then you can throw away your notion of high fidelity life. I think that comes when you have species which are sexually isolated and you can't have sex with other creatures because you're a eukaryote that's been sexually isolated because of your phylogeny. But if you were a bacteria, you would have no trouble having sex and exchanging DNA with other bacteria of different species. So there are many concepts that are associated with the last 2 or 3 billion years of the evolution of life, including information fidelity, that I think are just red herrings when it comes to trying to understand the origin of life.
Okay, thank you.
So another question? No?
You can't make your students ask questions?
Yes. Yes. Okay, so if not, let's thank Charlie again.
Okay. And